Welcome to the Science of Ageless Living. I'm Dr. Ethan Hausman-Marquis, doctor scientist, and the only peptide clinic physician in London with a published body of work specifically in this field. This is the Ageless Top 20 Peptides series. Every episode covers one compound, the real biochemistry, the honest evidence base, what we know, what we don't, and what the regulatory picture actually looks like. No hype, no discount codes, just the science. Today, SS-31. This episode sits in a different category from the last two. Cmax had real but structurally limited human data. Pinolon had almost no independently validated human data at all. SS-31, also known as Elamipretide, has something neither of those compounds has: more than 150 peer-reviewed publications, 2 decades of research, and as of September 2025, FDA approval. It is, in fact, the first mitochondria-targeted therapy ever to reach market. That changes how I need to frame this episode. We are no longer asking, is there any credible evidence here at all? We are asking a more mature and more interesting question. What does a mitochondria-targeted peptide that has actually cleared FDA review tell us about the broader hypothesis that mitochondrial dysfunction is a tractable target in human aging? And where does the approved indication end and the longevity extrapolation begin? Let's do it properly. To understand SS-31, you have to start with mitochondrial biology, and specifically with a molecule most people have never heard of, cardiolipin. Mitochondria are not simple, homogeneous organelles. The inner mitochondrial membrane is folded into structures called cristae, deep invaginations that dramatically increase the surface area available for the electron transport chain, the series of protein complexes that ultimately generate ATP. The structural integrity of these cristae depends heavily on a specific phospholipid found almost exclusively in the inner mitochondrial membrane, cardiolipin. Cardiolipin is not a passive structural component. It is intimately associated with the protein complexes of the electron transport chain, complex 1, complex 3, complex 4, and ATP synthase all require cardiolipin for proper assembly and function. It stabilizes the curvature of the cristae membrane, and critically, when cardiolipin is damaged through oxidation, through peroxidation by reactive oxygen species, the consequences cascade through the entire mitochondrial system. Cristae fragment, electron transport, efficiency falls, ATP output declines, and, in a particularly important downstream consequence, oxidized cardiolipin facilitates the release of cytochrome C from the mitochondria, which is one of the central triggers of apoptosis. This is where mitochondrial aging biology becomes directly relevant to this series. Cardiolipin content and integrity decline with age across multiple tissue types, and this decline correlates with cristae fragmentation, reduced oxidative phosphorylation capacity, and increased oxidative stress. A self-reinforcing cycle, since damaged mitochondria themselves produce more reactive oxygen species, which damages more cardiolipin. This cycle is one of the most consistently replicated findings in the biology of cellular aging, and it is implicated across an unusually broad range of age-related conditions: heart failure, sarcopenia, kidney disease, neurodegeneration, and age-related macular degeneration all share this underlying mitochondrial signature to varying degrees. The origin story of SS-31 itself is worth telling because it is genuinely unusual. The peptide was discovered, in essence, by accident in a pharmacology lab at Weill Cornell Medical College. In research led by Hazel Sito and her colleague, the compound was developed by Hazel Sito and Peter Barth at Weill Cornell. The original research program was not specifically hunting for a cardiolipin-binding peptide. The mitochondria targeting properties emerged from a broader investigation into small cationic peptides and their interactions with cell membranes. The serendipitous discovery that this particular peptide concentrated specifically in the inner mitochondrial membrane rather than distributing generally across cellular membranes was the finding that redirected the entire research program toward mitochondrial medicine. SS-31 is a synthetic tetrapeptide, 4 amino acids. Its sequence is DRGDMT lyse, Phe, and H2, and 2 structural features in that sequence are doing essentially all of the mechanistic work. The first is the alternating pattern of aromatic and cationic, positively charged, amino acid residues. This alternating arrangement is the key to the peptide's behavior at membranes. The second, more unusual feature is the inclusion of DMT, 2'6'dimethyltyrosine, an unusual, non-standard amino acid that is critical for the peptide's mitochondrial targeting properties. What does this structure actually achieve? SS-31 is able to selectively concentrate in the inner mitochondrial membrane at concentrations over 1,000-fold higher than in the surrounding cytoplasm. This is an extraordinary degree of subcellular targeting for a peptide that is administered systemically, and it is the central reason SS-31 has attracted sustained pharmaceutical interest. Most drugs intended to act on mitochondria struggle to achieve meaningful local concentration at the actual site of mitochondrial dysfunction. SS-31 essentially solves that delivery problem through its intrinsic chemistry rather than through a separate delivery vehicle. The proposed mechanism, in the most current mechanistic reviews, runs as follows. SS-31 selectively binds Cardiolipin on the inner mitochondrial membrane. This binding interaction stabilizes mitochondrial crystal structure, reduces oxidative stress, and enhances ATP production. Mechanistically, the cardiolipin binding interaction is thought to protect cardiolipin from oxidative damage, essentially shielding it from the reactive oxygen species that would otherwise peroxidize it, while also preserving the curvature and structural integrity of the cristae membrane that cardiolipin's correct conformation supports. This action helps optimize electron transport chain function, reduce reactive oxygen species generation at the source, prevent cytochrome C peroxidase activity, and restore crystal structure. The cytochrome C peroxidase point is mechanistically important and worth unpacking. When cardiolipin is oxidatively damaged, it can adopt a conformation that gives cytochrome C, normally an electron transport protein, a secondary pathological enzymatic activity where it catalyzes further peroxidation of cardiolipin itself. This creates a vicious cycle that ultimately drives mitochondrial permeability transition and apoptotic cell death. By stabilizing cardiolipin's native structure, SS-31 is proposed to interrupt this cycle at its origin. I want to flag the same honesty point I raised in the Pinolon episode because it applies here too, even for a compound with a far stronger overall evidence base. Despite abundant evidence for the broad therapeutic potential of SS peptides, the molecular mechanism of action of these compounds is, in detailed biophysical terms, still poorly understood. Recent biophysical work has been specifically aimed at characterizing exactly how SS-31 interacts with membrane surface electrostatics at a level of resolution beyond the general cardiolipin binding model. The broad strokes of the mechanism, cardiolipin binding, cristae stabilization, reduced oxidative stress, improved ATP output, are well supported. The precise biophysical detail of that interaction is still an active area of investigation, which is a more favorable position than Pinolon's largely unvalidated epigenetic hypothesis, but is still short of full mechanistic resolution. Now the evidence, and here's where SS-31 genuinely distinguishes itself from the 2 previous episodes in this series. Preclinical breadth. SS peptides have been shown by many independent groups using cell culture and animal disease models to have significant efficacy in restoring mitochondrial function across a remarkably wide range of pathologies: cardiomyopathy and heart failure, skeletal muscle injury and atrophy, ischemia and ischemia reperfusion injury, kidney injury and disease, neurodegenerative diseases, cancer, and the heritable mitochondrial disease, Friedreich's ataxia. This is a critical point of contrast with both Cmax and Pinolon. The preclinical work on SS-31 has been conducted by many independent laboratories, not concentrated within a single originating research group. That independent replication is exactly what was missing from the Pinolon evidence base, and it materially strengthens confidence in the core mechanism. Barth syndrome, the approved indication. This is the area with the strongest, most rigorously generated human data, and it's worth understanding why. Barth syndrome is a rare X-linked mitochondrial disorder caused by mutations in the TAZ gene, which encodes an enzyme called tafazin that is essential for proper cardiolipin remodeling. Patients with Barth syndrome have structurally abnormal cardiolipin from birth, leading to cardiomyopathy, skeletal myopathy, and profound muscle weakness. It is, in a sense, the most direct possible test case for a cardiolipin stabilizing therapy, a disease where the core molecular lesion is cardiolipin abnormality itself. SS-31 advanced through the Power series of clinical trials for Barth syndrome, and the regulatory journey was genuinely difficult. The manufacturer, Stealth Biotherapeutics, submitted its first new drug application for the Barth syndrome indication in August 2021, and the FDA refused to file it. A second submission in January 2024 secured priority review, but the FDA issued a complete response letter, essentially a rejection, in May 2025, while suggesting a path to accelerated approval based on muscle strength as an intermediate clinical endpoint. Stealth resubmitted in July 2025 and received accelerated approval on September 19, 2025. The approval was specifically for Barth syndrome with the brand name Forzinity, and the approved indication was narrow, specifically improving muscle strength in this rare disease population. I want to be precise about what that regulatory history tells us. This was not a smooth, first-pass approval built on overwhelming efficacy data. It was a long, difficult regulatory process with an initial refusal to file and a subsequent rejection that ultimately succeeded on the basis of an intermediate endpoint, muscle strength, rather than the broader functional or survival endpoints that might be considered more clinically definitive. That is a real approval by a real regulatory agency after real phase 3 level trials. It is not, however, the kind of slam-dunk efficacy signal that would let me wave away the narrowness of the indication. Heart failure. This is the indication most directly relevant to aging more broadly, since heart failure with preserved ejection fraction has a well-established mitochondrial dysfunction component that worsens with age. SS-31 has progressed through Phase 2 and Phase 3 trials for heart failure under the Triage HF program. Clinical trials in heart failure populations have shown improvements of 10 to 15% in peak oxygen consumption, VO2, after 4 weeks of intravenous administration, VO2 max being a well-validated functional measure of cardiovascular and mitochondrial capacity, and a meaningful clinical endpoint in heart failure research. This is a genuinely encouraging signal, though I'd note this particular figure comes from a secondary source rather than the primary trial publication, and the heart failure program has not yet, to my knowledge, resulted in an approved indication. It remains in active clinical development. Age-related macular degeneration. SS-31 has also advanced through clinical trials for dry AMD under the RECLAIM program, reflecting the high mitochondrial density and metabolic demand of retinal photoreceptor and retinal pigment epithelial cells, which makes AMD a biologically plausible target for a mitochondria-stabilizing therapy. This program, again, remains in active clinical development rather than at the approval stage. Mitochondrial myopathy and broader aging applications. Multiple other mitochondrial indications, including mitochondrial myopathy, are in active clinical development, and the preclinical literature extends further still into models of spinal cord injury, where Elamipretide has shown neuroprotective effects in both in vitro and in vivo models through mechanisms involving reduced pyroptosis and lysosomal membrane stabilization in injured neural tissue. Safety profile. Across this trial program, the safety profile has been described as favorable, with mild injection site reactions and headaches as the most common adverse events. The broader literature on SS peptides has consistently underscored their safety and favorable pharmacokinetic profile across the range of disease models and clinical trials in which they've been studied. This is a genuinely reassuring safety signal, generated across a substantially larger and more rigorous trial program than either of the two compounds in the previous two episodes. Here is where I need to draw the line precisely, because this is the crux of the longevity question for SS-31. The approved indication is Barth syndrome, a rare, genetically defined, pediatric mitochondrial disease with a specific molecular lesion in cardiolipin remodeling. The heart failure and AMD programs are in active phase 2/3 development, not yet approved. There is no approved or late-stage trial program specifically targeting healthy aging, age-related sarcopenia, or general mitochondrial decline as a primary indication in an otherwise healthy older population. The mechanistic logic for extrapolating from Barth syndrome to general aging is genuinely more direct than the extrapolations I asked you to scrutinize in the CMAX and Pinolon episodes. Barth syndrome's molecular lesion, damaged, improperly remodeled cardiolipin, is mechanistically continuous with the age-related cardiolipin damage I described at the start of this episode, even though the underlying cause is completely different. A genetic enzyme deficiency in one case, oxidative accumulation over decades in the other. The damage at the level of the cardiolipin molecule and the Cris-die structure it supports has meaningful overlap, but mechanistically continuous is not equivalent. Cardiolipin abnormality from a genetic enzyme defect present since birth in a pediatric population is a different biological starting point than oxidatively accumulated cardiolipin damage in a 65-year-old with otherwise normal Tafazin function. Whether the magnitude of benefit, the appropriate dose, or even the directionality of benefit transfers cleanly from one context to the other is an empirical question that the existing trials, focused on Barth syndrome, heart failure, and AMD, do not directly answer. The honest gaps for SS-31, even accounting for its considerably stronger overall evidence base, are still substantial when the question shifts specifically to longevity and healthy aging. There is no completed trial in healthy aging adults with mitochondrial function, biological age, or general functional health span as a primary endpoint. The approved indication and the late-stage pipeline are all defined disease populations: Barth syndrome, heart failure, AMD. The extrapolation to generalized age-related mitochondrial decline in an otherwise healthy person has mechanistic logic behind it, but no direct trial support. The optimal dose and route for a longevity application are not established by the approved data. The Barth syndrome approval and the heart failure trials use specific intravenous and subcutaneous protocols developed for those disease contexts. Typical research doses cited in less formal contexts range from 0.1 to 2 milligrams per kilogram daily subcutaneously, with intravenous infusion protocols in formal trials in the range of 0.01 to 0.25 milligrams per kilogram per hour over a 4-hour infusion. These are disease context doses, and translating them to a putative longevity protocol in a healthy adult is not something the trial data directly supports. The durability of benefit beyond the trial period studied, typically weeks, in the heart failure VO2 data is not well characterized for chronic, years-long use in an aging context, and the regulatory difficulty of the approval process itself is informative. An initial refusal to file, followed by a complete response letter on the second submission, followed by an eventual accelerated approval based on an intermediate endpoint rather than the most clinically definitive outcome measures. This is not the regulatory profile of an unambiguous, overwhelming efficacy signal. It's the regulatory profile of a biologically plausible, mechanistically well-supported therapy with a genuine but real and not enormous effect size in a disease population where any effective treatment was badly needed. Here is where SS-31 sits in 2026. It is a synthetic, mitochondria-targeted tetrapeptide with an unusual non-standard amino acid, DMT, central to its function that achieves extraordinary subcellular concentration at the inner mitochondrial membrane and binds cardiolipin to stabilize Cris-die structure, reduce oxidative stress, and support ATP production. It is the first mitochondria-targeted therapy ever to reach FDA approval, backed by 2 decades of research and a genuinely large and independently replicated preclinical literature. It has a real FDA-approved indication in a rare pediatric mitochondrial disease where the molecular lesion is directly relevant to its mechanism. It has active, late-stage clinical trial programs in heart failure and age-related macular degeneration, both conditions with substantial relevance to general aging biology and both showing encouraging, though not yet definitive, efficacy signals. It has a consistently favorable safety profile across a trial program considerably larger and more independently scrutinized than either Cmax or Pinolon. What it does not have is a completed trial or even an active late-stage trial, to my knowledge, in healthy aging adults using mitochondrial function or biological age as a primary endpoint. The longevity application remains, as with the other compounds in this series, a mechanistically coherent extrapolation from disease population data rather than a directly demonstrated effect. What distinguishes SS-31 from the previous 2 episodes is not that the extrapolation gap has disappeared. It hasn't. But that the foundation it's being extrapolated from is dramatically stronger. Independently replicated preclinical mechanism, multiple completed phase 2/3 human trials, 1 genuine regulatory approval, and active late-stage programs in conditions with direct relevance to aging. That is a different category of compound than one resting entirely on a single research group's unreplicated preclinical data. If you are a researcher or physician thinking seriously about which mitochondria-targeted compounds deserve attention in the aging space, SS-31 is, on the current evidence, the most clinically validated entry in that category by a considerable margin. If you are a patient being offered SS-31 specifically for longevity or anti-aging purposes outside of its approved or actively trialed disease indications, the question to ask your prescriber is the same one I've asked in every episode. Which specific human trial in which population supports the claim being made to you? And is that trial population actually comparable to you? If this episode has been useful, share it with someone navigating this space, particularly anyone being offered mitochondrial peptide protocols without a serious conversation about the difference between an approved disease indication and a longevity extrapolation. This field deserves more precision and less noise. In the next episode, we'll be covering TB500, one of the most widely used peptides in the longevity and performance clinic world, as it's traditionally stacked with BPC157, and perhaps the compound where the gap between the evidence people believe exists and the evidence that actually exists is largest. We'll do it the same way. Mechanism, evidence, honest gaps, no shortcuts. I'm Dr. Ethan Hausman Marquis. This is the science of ageless living.